Hello. Today we're going to be going over the endomembrane system. This is the second lecture in the cell organelles. So with the endomembrane system, hopefully you know endo means inside, membrane, obviously. But the main component of these is endoplasmic reticulum. Now there's two kinds of endoplasmic reticulum. There's a rough and a smooth, and the major difference between the two is that the rough endoplasmic reticulum has ribosomes on it, and the smooth endoplasmic reticulum doesn't, and therefore it changes kind of how they work and what they actually do within the cell. Now in general, these two kind of help move substances within the cells. It's almost, you can picture them almost like the intestines. They can help move things around. There's a bunch of them. They can either be straight in some cells, or they can be an interconnected membrane, which has a bunch of different doors and hallways, and goes in a bunch of different directions, almost like a web. Now the rough endoplasmic reticulum, or the rough ER, the ribosomes attached to the surface. So as you can see down below, you can see the little ribosomes on the actual surface of this membrane. Now it's usually wrapped around the nucleus, as you can kind of see in this picture. That's a common place for it. Now what they're really doing, or what this rough endoplasmic reticulum is doing, is kind of doing two things. Kind of gives ribosomes a home. That's one. It kind of gives a central base to it, and a central place for all the ribosomes to do the proteins, and also helps modify the ribosomes, or helps modify the proteins, with the ribosomes. So this is actually a recent discovery. For a long time we just thought it was kind of just a home for the ribosomes to be, a central area where they could do work. Yet more and more we're finding out that the rough ER kind of does a little bit more with the proteins than we originally thought. Alright, the smooth endoplasmic reticulum has no ribosomes attached, and they kind of are the building center. They build some of the molecules that you find in the cell, things like carbohydrates and lipids. Now in the picture over here, you can see the smooth endoplasmic reticulum over here. In some cells and some diagrams, you'll see the smooth endoplasmic reticulum actually connected to the rough endoplasmic reticulum, but in this picture they decided against that. It depends on the cell if that is actually the case, but really the big difference is no ribosomes, and you're building molecules here, and these enzymes are the things that help build them. The Golgi apparatus is actually a really interesting organelle, and you could also call it the Golgi body. It just depends on where you learn it. Now these are really, depending on the kind of cell, they have different jobs, and in plants they're involved in the plant cell wall creation. So they're help building parts to create the cell wall. Obviously animal cells do not have cell walls, but in animals and in plants the other main job is it's the packaging and shipping station of the cell. Most things that are leaving the cell have to go to the Golgi apparatus. The Golgi apparatus puts a little post tag on it, or a little stamp, and it tells that object, like a protein, where it needs to go. So it will go out to the end of the cell membrane, leave the cell, and that little post tag will tell it where it needs to go in other cells. Now, to explain this a little bit better, first it usually involves vesicles. We'll talk about what a vesicle is in a second, but molecules come in with vesicles. These vesicles fuse with Golgi membrane. The molecules get a little stamp on them in the Golgi. This vesicle leaves, and then the vesicle goes to the plasma membrane, and the contents are let go out of the cell. So here's the picture. So we got a vesicle here. It has some content. Goes to the Golgi apparatus, gets a little modified cell, gets into another vesicle, and then leaves and connects to the cell membrane and goes out by something called exocytosis. Lysosome is one of the interesting ones, and one that everyone typically remembers. It's basically the garbage center of the cell. That contains digestive enzymes that help break down different old organelles and different parts that the cell doesn't need anymore. This kind of helps in cell renewal. The cell recycles parts, because if it didn't, it would take so much energy just to remake all these things that the cell wouldn't be able to do other activities needed for life. It also can digest invaders, which is an important thing. If the cell realizes there's an invader, it'll put a wall around the invader, and then the lysosome will come over and kill the invader with its enzymes. Now, if a lysosome erupts inside of a cell, the cell will actually be eaten from the inside out. The enzymes will just kill everything, eat everything. So it's one of these things where it's not really, it doesn't have a brain, doesn't have a function, these enzymes, besides just destroying things. So the cell has to be really careful on what it releases these enzymes onto. If it releases it onto a mitochondria that's not supposed to be broken down, then that mitochondria will be completely eaten. Peroxisomes. Now, peroxisomes are something you didn't learn in regular biology. They're some new kind of organelle. It's very similar to the lysosomes, but it has a very different task, but very important task. Hydrogen peroxide is a dangerous, toxic chemical inside of your body. It's actually a poison. Now, we use hydrogen peroxide. You guys probably seen it. It's in a black bottle with a white top, and you get a cut, and your parents would have put it on there to kill bacteria and stuff like that. That's fine. But high amounts of hydrogen peroxide, especially in a small cell, will kill it. Now, to break down certain things, you actually need hydrogen peroxide in some of these organic reactions. So the peroxisomes create hydrogen peroxide, which helps break down lipids, but then it breaks down the hydrogen peroxide that it used. So it's like a contained little chemistry set in there where it breaks down things, but it doesn't hurt the cell. And then therefore the cell can break down things that if it did in the original cytoplasm or something like that, the cell would actually die. Vacuoles and vesicles, depending on the type. Vesicles, which isn't listed here, vesicles are just basically little storage centers. They're like little balls that your cells put different things into. It could be food, water, waste, whatever it needs. Vacuole is typically just found in plant cells. It's one big one, usually in plants. When I say big, it usually covers about 98% of the cell, so it is a very, very huge storage. And in one vacuole, you can actually contain all the contents, water, food, and waste. So you can kind of see a picture down there, right there, what it looks like. And that's the end of the presentation. Not a lot in this section. Make sure that you know the differences between them. Make sure you know the basic facts about them. And I will see you in class. Make sure you watch Part 3.